Hello, and welcome to our series on pathogen-specific considerations for investigating foodborne illness outbreaks. In this video, we will discuss specific considerations for investigating norovirus outbreaks. First, let's go over some of the basics of viruses. A virus is a type of microorganism that can only infect and grow inside their specific host. Outside of these living hosts, viruses will eventually die off. For example, some viruses, such as norovirus, can specifically infect and cause disease in humans. Outside of the human host, norovirus can only die off. However, because of its structure, this die-off can take a long time. Viruses are generally categorized as enveloped or non-enveloped based on their structural features. Some examples of enveloped viruses include influenza A virus and SARS-CoV-2, and some examples of non-enveloped viruses include norovirus and hepatitis A virus. Both enveloped and non-enveloped viruses have receptors that are needed in order to infect host cells. The main difference here is that the receptors on enveloped viruses are embedded in a weak lipid envelope, whereas the receptors on non-enveloped viruses are embedded in the strong capsid layer. Due to the presence of the strong capsid layer, most non-enveloped viruses, including norovirus, are resistant to many common sanitizing chemicals used on the market. For example, quaternary ammonium compounds, which is a common active ingredient in many cleaning wipe products, and alcohol, which is a common ingredient in most hand sanitizers, are not effective against norovirus. However, it is important to note that some chemicals, such as chlorine, is effective at removing norovirus from surfaces, and some activities, such as washing hands with soap and water, is effective at removing norovirus from hands. Additionally, the strong capsid layer can help norovirus survive other environmental stressors and persist on food-associated surfaces for long periods of time, sometimes upwards of 2 weeks. Therefore, it is important to ensure that the right chemicals are used for various activities, such as using soap and water to wash hands and using chlorine to wash food handling equipment in order to reduce risk of transmission of norovirus. Given these characteristics of norovirus, it is not surprising that this pathogen represents the leading cause of foodborne illness in the U.S., with 19 to 21 million cases occurring each year. Common symptoms of norovirus illness include diarrhea, vomiting, nausea, abdominal cramping, and fever. Generally, norovirus has a short incubation period ranging from 12 to 48 hours. This illness is typically self-limiting, with symptoms of norovirus typically lasting between 1 to 3 days. Common local establishments where norovirus outbreaks can occur include restaurants and other eateries, long-term care facilities, schools, cruise ships, and penitentiary facilities. When investigating norovirus outbreaks linked to local establishments, there are a few key areas where environmental health investigators should focus their efforts. These include evaluating whether infected food handlers may have been the source of the outbreak, and evaluating if cross-contamination of norovirus from infected food handlers to food products could have contributed to the outbreak. First, let's discuss how to evaluate whether infected food handlers may have been the source of the outbreak. To evaluate whether infected food handlers could be the source of an outbreak, environmental health investigators should first identify whether any food handlers or other employees at the establishment showed symptoms of foodborne illness. This can be done by reviewing relevant documents either during or prior to site visits, such as 1. Any available employee call-out records, 2. Sick leave policy documents, as well as 3. Other relevant documentation on recent illness reported by employees of the establishment. In addition to reviewing records, environmental health investigators should also conduct interviews with both upper management and food handlers within the establishment during investigational site visits. Some examples of important questions to ask in these interviews include 1. If and when any food handlers or other employees started showing symptoms of foodborne illness. 2. Whether there is an issue with short staffing or high rates of turnover. And 3. How sick leave policies are implemented and enforced. Gathering information on how sick leave policies are regularly enforced is especially important because if written sick leave policies are not appropriately enforced, the establishment could be at an increased risk for norovirus outbreaks. For example, in a 2017 norovirus outbreak associated with a restaurant in Virginia, interviews with key food handlers helped environmental health investigators learn that managers often pressured food workers to come into work even if they were showing symptoms of foodborne illness, which contradicted the restaurant's sick leave policy document. If one or more cases of foodborne illness are identified among food handlers or other employees based on record review and interviews, it is important to identify the exact dates in which these individuals reported showing symptoms, when they attended work, and how these dates correlate with estimated exposure dates for people affected by the outbreak. This information can help investigators identify whether the infected food handlers could potentially have been associated with the outbreak. For example, in this scenario epidemic curve of a norovirus outbreak, food handler A first reported showing mild symptoms of foodborne illness at work on both April 27th and April 28th, but did not call out sick from work until April 30th, when their symptoms became more severe. As we know that norovirus has a typical incubation period of 12 to 48 hours in which individuals may still be able to shed the norovirus without showing symptoms, mapping when food handler A started showing symptoms and when they stopped attending work to this scenario epidemic curve provides evidence to support that this food handler may have been a potential source of this norovirus outbreak. Next, we will discuss how to evaluate if cross-contamination of norovirus from infected food handlers to food products could have contributed to the outbreak. It is important to note that norovirus can be spread to food products from infected food handlers if they aren't frequently washing their hands or wearing gloves. And, in particular, foods that are handled after cooking or require a lot of manual preparation, such as chopped salads, deli sandwiches, or fruit platters, are at high risk for this type of cross-contamination. To identify whether food handling practices could have facilitated cross-contamination of norovirus from infected food handlers to food products, environmental health investigators should evaluate the practices used to prepare food products in the establishment during site visits. This can be done through observing how food is routinely handled in the establishment. During these observations, one key thing to take note of is the accessibility of hand-washing sinks and gloves in the establishments to promote frequent hand washing. Are hand-washing sinks properly stocked with soap and paper towels to promote proper hand washing? Are trash bins strategically placed near hand-washing sinks? Is the temperature of the water in hand-washing sinks appropriate to achieve effective hand washing? Are gloves available and easily accessible to food handlers? Additionally, attention should be paid to the behavior of employees with respect to their hand hygiene and glove changing practices. For example, how frequently are employees washing their hands or changing their gloves? Are employees washing their hands after using the restrooms? Are food handlers recontaminating their hands and gloves after washing by touching their face and other parts of the body? Are food handlers wearing gloves when handling higher risk foods, such as those that are not cooked prior to consumption? Additionally, investigators can also conduct interviews with both upper management and food handlers about these routine food handling practices. Important questions to ask during these interviews include 1. How is hand washing enforced in the establishment? And 2. Are food handlers encouraged to wash their hands or change gloves between food handling activities? 3. Are there instances in which food handlers are advised against using hand-washing sinks or changing gloves due to scarcity of the supplies? These questions are all particularly important to ask, as any practices that deter food handlers or other employees from washing their hands or changing their gloves can potentially contribute to the spread of norovirus from infected food handlers to food products. For example, in a 2022 norovirus outbreak associated with a restaurant in Illinois, environmental health investigators conducted a site visit to investigate the outbreak, where they observed a food handler handling food items at a salad bar without wearing gloves. The same food handler reported showing symptoms of foodborne illness, like vomiting, at work during the week in which all cases associated with the outbreak were reported. In summary, in this presentation, we have covered some of the specific characteristics of norovirus, as well as the two key areas that are relevant for environmental health investigators to consider when investigating norovirus outbreaks linked to local establishments. In addition to norovirus, check out our other videos in this series that cover key considerations for investigating foodborne illness outbreaks linked to other foodborne pathogens.